This video will focus on DNA replication. After watching the video and engaging with the material presented, you should be able to explain some of the early experiments that demonstrate that DNA is indeed the genetic material in living organisms, and you should also be able to explain the basic mechanism behind DNA replication. Let's begin with recognizing that up until the 1940s, early 1950s, we really didn't know for sure what the genetic material was in living organisms. We knew that whatever it was located in the nucleus, but the nucleus contains lots of things. There's proteins, RNA, carbohydrates, DNA. So which of those molecules actually serves as the genetic material? And how do we know? Well, of course, it's common knowledge these days that it's DNA, right? That's the genetic material. But how do we know? Well, just a few experiments early on that led to this conclusion that it's DNA and not any of those other types of molecules that serves as the genetic material. Let's begin with Griffith's experiment. Griffith was doing his work in the 1920s. He was using a bacterium known as Streptococcus pneumoniae. You might guess from its name that this bacterium can cause pneumonia. And it turns out that there are two strains that he was working with, a smooth strain toward the left and a rough strain toward the right. Turns out the smooth strain produces this outer capsule, whereas the rough strain does not. And the smooth strain basically contains genes, we now of course know, that are responsible for coding for that outer capsule, again just shown schematically in green in the image on the screen. So again, he's working with these two strains of bacterium, and he essentially recognized that these two strains have different levels of pathogenicity. So for example, if you take rough strain cells, inject them into mice, the mice can survive that. So the mice live, no problem. So we say, well, the rough strain is benign. On the other hand, the smooth strain, if you inject that into mice, the mice die. So the smooth strain is virulent. But then he wondered, what if he boiled? What if he killed the smooth strain bacteria and then injected those into mice? Well, it turns out the mice lived just fine. So the boiled or heat-killed smooth strain are benign. But then what's interesting, he thought, well, what if he took heat-killed smooth cells, so ones that he had boiled and killed them, mixed those with living rough strain cells, and then injected that mixture into mice. So given that setup, what do you think would happen toward the right here in this situation? After all, we established earlier that the heat-killed smooth strain had really no impact on the mice. It was benign. The rough strain, same thing. It was benign. Mice could live after being infected. But when you mix the two, you might predict, well, the mice would probably live, no problem. But it turns out they died. Not only did the mice die, but you also found that they were loaded with living smooth strain bacteria. Where did that come from? After all, we didn't inject the mice with that, but it turns out there was something, some molecule that was present in these boiled smooth cells that made its way into the living rough cells and therefore transformed them into the smooth strain. So what was it that caused that transformation? Well, of course, we now know that it was DNA. It was some genes that codes for that outer capsule. But Griffith had no idea. He didn't know. Others didn't know either. But there was something, right? Some genetic material, right, that could transform those bacterial cells. So Avery, McCarty, and MacLeod kind of picked up on Griffith's initial work and addressed this basic question. Is it protein that's the genetic material? Is it DNA? Is it RNA? Using the same experimental system that Griffith did earlier. And their logic was as follows. They took heat-killed smooth cells and then separately, in 3 separate batches here, added enzymes that broke down all the proteins, but left the RNA and DNA intact. To another batch, again, they took heat-killed smooth cells, added enzymes that broke down all of the RNA, but left the protein and DNA intact. A third batch, same idea, but they had enzymes that broke down all the DNA and left the proteins and RNA intact. Then they mixed each of those with living rough cells and then asked the question, okay, which, if any of those treatments results in these rough cells being transformed into smooth cells? Sure enough, what they found was if you break down all the proteins, that's okay. Transformation still occurs. Break down all the RNA, that's okay too. Transformation still occurs. But when you break down the DNA, transformation is not able to take place. So in other words, you can't convert these living rough cells into the smooth strain. That's evidence right there that it's DNA, right, that serves as the genetic material, at least in this particular situation. So there were a variety of other experiments that led to this conclusion that indeed it's DNA that serves as the genetic material. So we now know that DNA is a genetic material, and we know that when cells divide, they have to somehow give exact copies of all the DNA, of all the chromosomes, to the newly formed cells. So what occurs during DNA replication as this occurs during S phase, or the synthesis phase, of the cell cycle? So just a quick review of the cell cycle. Shown schematically here, let's say you have a cell that was just formed by mitosis. That cell will exist for a period of time, the so-called G1, or first gap. It's doing its thing, whatever it's supposed to do. But then if this cell itself is going to eventually divide by mitosis, we have to replicate all of the DNA, all the chromosomes in that cell, so that each of the newly formed cells gets a complete set of chromosomes. So here's the S phase of the cell cycle. This is where DNA synthesis occurs. That's then followed by some period of time, the G2 phase, or the second gap. And then when conditions are just right, that cell can then itself undergo mitosis. So really this whole chapter is taking a look at this part right here, DNA synthesis, what takes place during that phase. If, for example, I were to ask you to draw a chromosome, you would probably come up with something like you see up on the screen here, maybe something simplified like these first two, or maybe a little more fancy like this one down here. But these are all viable chromosomes. But notice each one of these consists of two chromatids. I just want to point out that if you draw a chromosome that looks like either one of these that consists of two chromatids, that's what they look like after DNA synthesis has taken place. Prior to DNA synthesis, a chromosome might look just something like this, just basically one piece of DNA. Yes, double-stranded, yes, double helix, but really just one chromatid, if you will, just one piece of DNA. So the whole goal of this chapter is to see, okay, how do we go from a chromosome that's just one piece of DNA to two identical pieces of DNA, these two chromatids, right? That's the whole point of this whole chapter. Okay, so how is DNA actually replicated during the S phase of the cell cycle? Well, there's early work by Messelson and Stahl. They performed a classic, elegant experiment demonstrating that DNA replication is semi-conservative in nature. And by that, all we mean is if you have an original piece of DNA, shown here in gray, what happens during DNA replication is both strands separate. So the hydrogen bonds are broken toward the middle. Each strand, again shown here in gray, then serves as a template so that new complementary bases can be added to each of those templates. And we end up with two pieces of DNA which are genetically identical to each other and to the piece of DNA that we started with. And again, keep in mind these two pieces of DNA here, we're sort of zooming in on two chromatids of a chromosome. So that's what we're going to look at in just a little more detail. So let's look at the mechanism of DNA replication. Think back to the basic structure of DNA. It's focused toward the left here. You may recall the 5' and 3' orientation of each side or each strand of DNA. So this is important. Now we have to focus a little more on the 5s and the 3s because the location and the orientation of those is crucial to understanding everything that occurs. And so, for example, when a new piece of DNA is being synthesized, complementary bases are added only in a 5' to 3' direction. And just keep in mind that refers to the direction of the newly synthesized strand. Can get a little confusing. So just a reminder, if we look here, this is part of a DNA molecule. Here is a 3' carbon right here. So here's a 3' hydroxyl group. The 5' end would be up here, the opposite end. But a base, when it's added, is linked onto this 3' hydroxyl. So the point is, when you're adding new DNA bases, they're always added in a 5 to 3 direction. So you have to keep that in mind. That's crucial. Another thing to keep in mind is that we actually have to form a short primer, which is typically a short piece of RNA, before we can add new DNA bases. So this is shown schematically here. So let's initially focus on this top portion here. Imagine a piece of DNA. It's unwinding. The two strands are separating, let's say, in a left to right direction in this particular case. As the two strands unwind, one of the first things that gets produced is this short primer. So shown in yellow here, as well as here. Again, that's a piece of RNA. So we have two RNA primers indicated here. Once those small pieces of RNA are added, and again, of course, their bases are complementary to the template, then, shown here in this reddish color, then DNA bases are added, again, any 5 to 3 directions. So again, keep your 5s and 3s organized here. So original DNA, if this is your 3' end, this, of course, would then be the 5' end. The newly synthesized DNA is added in a 5 to 3 direction, again, of the newly synthesized strand. So what you notice here is that if we take a look at this top strand, again, imagine this continues to unravel, unwind in a left to right direction. We can just continue to add new DNA bases, right, complementary to the template strand, in a 5 to 3 direction, at least on this top strand. However, because of this 5 to 3 constraint here, think about what happens on this bottom strand. Yes, we opened this up and we added an RNA primer here, and that can be used to synthesize new DNA, again, in a 5 to 3 direction of the newly synthesized strand. But notice on this bottom strand, that creates a potential problem, because now we have this sort of gap here. We have to deal with that. We have to contend with that. We will in just a moment. I just want to point out that it turns out that we can identify the so-called leading strand. It's this shown here. It's the top strand. DNA synthesis on a leading strand is continuous, and it forms essentially a nice, long, continuous piece of DNA. However, this strand down here would be considered the lagging strand, because DNA replication on this strand is discontinuous. And what we end up doing is forming small fragments, so-called Okazaki fragments, named after the person who sort of discovered these, that will then live. So this is one Okazaki fragment right here, but we'll produce a bunch of these Okazaki fragments on this lagging strand, which will later be connected to give us 2 complete pieces of DNA. Okay, so just a few of the proteins and enzymes involved in this whole process. We're not going to mention all of them. We'll just mention a few. First of all, we have to somehow unwind or separate the 2 strands of DNA. So that's what helicase does. So helicase comes along and separates the 2 strands of DNA. Then we want to prevent those 2 strands from snapping back together. So that's what these single-strand binding proteins do, these SSBPs. They prevent the 2 strands from snapping back together. And then we want to produce this little piece of RNA that can serve as a primer. So the enzyme primase does just that. So shown here in yellow, produces this small sequence of RNA, again, complementary to the sequence on the template DNA strand itself. So now everything is set. DNA polymerase III can come along, and it can add DNA bases, again, in a 5 to 3 direction. So that's what's illustrated here, this green molecule, that's DNA polymerase III. The DNA bases are shown here in red. And then if you think back to the Okazaki fragments formed on the lagging strand, we have to deal with those before all is said and done. So a few more enzymes we'll mention here. DNA polymerase 1. This is the enzyme shown here in yellow that comes along and removes those RNA bases and replaces those with DNA bases. So that's good. You don't want to have a bunch of RNA interspersed in your newly synthesized DNA. So you get rid of the RNA bases, replace those with DNA bases. But even at that point, you still have a little gap between your Okazaki fragments. So there's one more enzyme we'll mention, namely DNA ligase. This comes in, forms covalent bonds between adjacent DNA bases, thereby completing the process. So as long as everything is working correctly, we end up with two genetically identical pieces of DNA. But mistakes can happen. There are all sorts of things that can lead to various mutations and so forth. So mistakes can happen that might have some implications or might not. It just depends. But just a few things we'll mention here is that the enzyme DNA polymerase 3, it's pretty good at catching potential mistakes. So imagine all the trillions of cells in your body. When they're formed by mitosis, or if they themselves undergo mitosis, you have to replicate all the DNA, all the billions of base pairs, right? You have to have enzymes that are working pretty good and not making too many mistakes. That could lead to mutations. So DNA polymerase 3 can proofread. It can catch mistakes that it might make. So for example, if it accidentally adds a C opposite an A, well, that's not right. So it can pause for a split second, catch that mistake, kick the C out, and replace it with the appropriate base. So it's pretty good at correcting its mistakes and preventing them from being incorporated into newly formed DNA. But again, not always. Mutations can occur. But also another concept I just want to mention is that often we can have bases, DNA bases, that can become damaged in a variety of ways. For example, think about ultraviolet light shining on cells that contain DNA. Well, it turns out often if you have two thymines, so two T's adjacent to each other in DNA, when they're exposed to UV light, and this happens all the time in all of our bodies, the so-called thymine dimers can form. So you get these bonds formed between the two thymines. That causes a change in the shape of the DNA. That could be very problematic. But it turns out in most of us, I mean, not everyone, but most of us, fortunately, there are a whole series of proteins and enzymes that can recognize this problematic region of DNA. So can recognize thymine dimers like we see here, remove the whole problematic area, replace those with normal bases, and thereby prevent any potential problems. So we do have protective mechanisms in place, fortunately, that can deal with damaged bases. But again, they don't work all the time, right? Mutations can occur, they can persist, and of course, they can potentially have very serious implications. So that concludes our brief discussion of DNA and the gene, synthesis, and repair.